So metabolomics is a set of emerging technologies that have been developing for the last 10, 15 years or longer that enable us to measure large numbers of small molecules in a single sample of blood or some other biological sample. And the advantages of that are numerous. It was developed to some extent as a way to complement the significant advances that have been made in genomics to be able to look at large numbers of genes and variations in genes, variations in expressions of genes, looking at large numbers of proteins. It's one of those types of so-called omics technologies that allow us to get a very, very large amount of data on a large number of very important molecules in the same sample. And there are many advantages to that, because then you can start to look at interrelationships between those molecules and their relationships to genes, proteins, and other processes. Metabolites really are in some ways the workhorses of cells and of our bodies. They're the molecules that actually do the things that are directed by us. So our genes tell our cells to do certain things, and that they do that through translation into messenger RNA, or transcription into messenger RNA, translation into proteins, and then the proteins operate on, to a large extent, on these small molecules and do various things. Glucose being a small molecule, for example, and a whole amino acids that are used by our bodies all the time in producing proteins and to create signaling molecules and so on. There's a substantial number of these molecules that are readily measurable in these biological fluids, and that by measuring many of them, we can make inferences about what's happening at the level of enzymatic reactions or other metabolic processes.